In the cell, chemical reactions don't happen in isolation. They'll occur in complex metabolic pathways that put reactions in order, transitioning from one product, or where the product becomes the starting material for the next reaction. So if I look at this example, the goal is to have a final product that has 3 phosphates on it, and my starting material has no phosphates on it. And the way that the cell will accomplish this is by 3 chemical reactions, adding the phosphates in 3 distinct steps. The product of one reaction, so if I just label these A, B, C, and D, A will make B, but then the next reaction will take B, which is the product there, and make it a reactant. B becomes C, and then C becomes D. So 3 distinct reactions, but they flow into each other because the product of one reaction becomes the reactant of the next. And so, again, in the cell, this is how all of the chemical reactions happen, or in long sequences like this. And so in the next chapter, for example, we'll begin to look at glycolysis, for example, which is 10 chemical reactions that happen in order like this. Metabolic pathways can be broken into 2 broad categories, or classified in 2 broad categories. Catabolic, which are pathways that break materials down and are typically exergonic, or releasing energy. Anabolic pathways, on the other hand, are making things, so not breaking them, but making them, and are endergonic. Because they're endergonic, they'll have to be coupled to an exergonic reaction. Remember, endergonic means we need an input of energy. That energy has to come from somewhere, and so an exergonic reaction can provide that energy. So again, catabolic reactions are the exergonic breakdown of reactants, and they're going to release energy that can be used for other things. Catabolic reactions can also recycle or break down materials that can be reused, and we saw that when we were looking at the lysosome in the cell chapter. Here we're going to focus on what happens to the energy that's released. So when this energy comes out of these exergonic reactions, it will be immediately put into energy intermediates. Energy intermediates are stores of energy. And we're going to focus on ATP and NADH. So exergonic reactions will put that energy that they release into ATP and NADH, which can then later be used by the cell for endergonic reactions. So ATP and NADH are now what we're going to focus on. So ATP can be made in 2 ways, and we'll see this in the cellular respiration. In one way, it's called substrate-level phosphorylation. It starts with a molecule that has a phosphate on it. So let me put this up here to show you the chemical reaction differently. There's a phosphate here, and it's going to combine with ADP, and then it's going to produce the substrate without the phosphate and ATP. So in substrate-level phosphorylation, the phosphate on one molecule comes off, so this bond breaks. The energy of that is used to put the phosphate onto ADP, which makes ATP. So in essence, the phosphate is transferred, and there's energy in that transfer that can put energy into ATP. Chemiosmosis is actually the way the majority of ATP is made in the cell, and this is when gradient energy, so remember we said a gradient is a form of energy, can be used to take, so if I write that chemical reaction, it takes ADP and a free phosphate and makes it into ATP, and the gradient energy provides that energy. So we'll focus more on this one in a later chapter. NADH is the other energy intermediate, and it's made in a very different way. It's made through a kind of reaction, or a group of reactions called redox reactions. Redox stands for reduction, oxidation. So they always go together. If there's a reduction, there's an oxidation. So that's where redox comes from. So an oxidation is when electrons are removed, and those electrons must go somewhere, and that's the reduction part. So the electrons that are removed are added to something else. So that's why they always go together. One thing loses electrons and one thing gains electrons. We saw this before when we looked at ion formation. Sodium loses electrons and chlorine can gain those electrons. Putting that together is a redox reaction. One is oxidized, the other is reduced. So more generally, we have A losing an electron, so it is being oxidized to A, and B is gaining an electron, so it is being reduced to B with the electron. Right? So always they go together, a reduction and an oxidation. One thing is oxidized, one thing is reduced. NADH is created this way. So NADH is created when NAD plus is reduced to NADH. So remember, that means it's taking electrons. So you can sort of remember this by seeing how the positive sign goes away. So it's positive and it's not positive, so that means the electrons must have gone in. But remember, if electrons went in, they must have been taken from something else. So some other organic molecule loses its electrons at the same time. And that's what is used to reduce NAD+. So the redox reaction, whenever you see NAD+ being reduced to NADH, you know there must be a redox reaction happening. Whatever other molecule is involved must be losing the electrons. The electrons are transferring energy, so we can kind of think of electrons as carrying energy. So when the organic molecule lost the electron, it also lost energy. And when it gave that electron to NAD+, it gave that energy to NADH. And so later on, NADH can release that energy. And again, remember, we said NADH is storing energy. It's storing energy in those electrons. So how are we going to identify redox reactions in biology class? Well, you're not really, haven't got the background in chemistry from my class to be able to identify these based on the chemistry of the molecules. And for our intents and purposes, we mainly are going to use information that we know about NAD+ to identify them. So we know that if we see NAD+ becoming NADH, so notice the direction of my arrow, then that must be a reduction. And because it must always go together, if NAD+ is being reduced, it must be getting those electrons from this molecule. We don't see it's whatever the product is, but there must be some other product here. So this must be being oxidized. If I look down here, NADH is going to being NAD+. So we said earlier that NAD+ going to NADH is being reduced. It's gaining electrons. To go the opposite direction, NADH to NAD+ must mean it's losing electrons, and that is an oxidation. So if going one direction is a reduction, going the other direction must be an oxidation. So if this is being oxidized, then this molecule must be being reduced. So a little different than what we saw. Sometimes people see these hydrogens and they get a little confused. The hydrogens are playing a role in this reaction, but they're neither being oxidized nor reduced. They're not participating in the redox part of the reaction. They're important, but they're not participating in the gain and loss of electrons. The last thing I want to say about this is that sometimes people have a hard time remembering which one is which. And this is kind of a silly little memorization device, but it's always worked for me, so I like to share it. Loss of an electron is an oxidation. Gain of an electron is a reduction. Leo the lion goes grr. Loss of an electron is an oxidation. Gain of an electron is a reduction. So hopefully that helps you remember these terms. And then also just reminding you that in biology, look for NAD. Whatever's happening to NAD or NADH tells us what is being oxidized and what is being reduced in the chemical reaction. And then again, too, remember that NADH is an energy storer. The energy that is in NADH can later be used by the cell.